Great. Well, welcome everybody here in the room and on Zoom. It's great to see such a full house for this event. My name is Kartika Pallar. I'm an associate professor and the chair of the UCSF Academic Senate Committee on Research. And I want to welcome you all here today. Before we get started, we'll just go over a few housekeeping tips. So there will be a Q&A session at the end of the program. For those on Zoom, please put your questions in the Q&A box at the bottom of the screen. And finally, this event is being recorded and will be available on the Senate's website. And now it is my great honor to introduce the UCSF Academic Senate's 67th Annual Faculty Research Lectureship in Basic Science. The Academic Senate Faculty Research Lecture started in 1957. The first lecture was given by Professor Melvin Calvin on the topic of photosynthesis as part of the Charter Day activities commemorating the birth of the University of California. Originally, an ad hoc committee was appointed to nominate the first faculty research lecturer. From 1960 on, the Committee on Research decided to take on this responsibility, and in 1966 decided to solicit new nominations from department chairs, deans, directors of institutes, and members of the Senate. Since then, nominations are made by UCSF faculty who consider the scientific research contributions of their colleagues and submit nominations for this prestigious award to the Academic Senate Committee on Research. I'd like to thank the committee for their hard work in selecting this year's nominees and winners. This Faculty Research Lecture Award recognizes exceptional career achievements in basic science research. And today, we are proud to honor our colleague, Dr. Richard Locksley. And now I am pleased to introduce Professor Jason Sister, who nominated Dr. Locksley for this award. He is an investigator at the Howard Hughes Medical Institute and professor and vice chair in the Department of Microbiology and Immunology. He will provide an introduction to Dr. Locksley.
Well, thank you, and thanks to the Academic Senate for holding this series of lectures celebrating basic science. Maybe before I get going, if people want to come down the front, there's a few more seats here, so feel welcome to come down. But as a nominator, and I think one of quite a few people here today who came to UCSF in part, at least because of Rich, it's really a pleasure to introduce him for this honorary lecture. And Rich is the Sandler Distinguished Professor of Medicine, investigator of the Howard Hughes Medical Institute, and director of the Sandler Asthma Basic Research Center. He was born in Minneapolis, but grew up in many places because he was in a military family, but found his way to Harvard and then on to Rochester for his MD before we recruited him here for his internship and residency. He then moved up to UW for his infectious disease fellowship and started there as an assistant professor before we recruited him back as a young chief in the Division of Infectious Diseases, a post he held for almost 20 years. And during that time, he started the first infectious disease training program here. And he was also an attending physician here for even longer. So really substantial contributions to our clinical enterprise. But today we're focusing on his contributions to basic science, which I think really got underway when he was an infectious disease fellow at UW. And his fascination at that time with the responses to different types of infections, and I think an early realization that different subsets of helper T cells would be contributing to different types of responses. But and I've been following Richard's work, I think almost since the outset, certainly since the early 90s, and I think I know it pretty well. But I was sort of worried, with more than 40 plus years of contributions, maybe I'd missed something important. So I thought I should ask Versa. And so I asked Versa, what is Rich Locksley at UCSF known for? And of course, within seconds, I got all this. But it's actually, I think, fairly accurate that Rich is known for his groundbreaking research in the field of immunology, particularly his work in the immune system's response to infections and the roles of cytokines in immune regulation. I think Versa got the order a bit wrong. The Th2 cell work really was the early work that delved into Th2 responses to different pathogens and helminths in particular. And that segued into more work on the cytokines involved and ways to illuminate these important communication molecules in the immune system. And that led to this seminal breakthrough work in the discoveries of innate lymphoid cells and really their profound contribution to launching and guiding many types of immune responses. And all that fed into Rich's contributions to allergy and asthma research and the potential for therapies that are coming from that. I would say that I think there's at least one important scientific contribution missing from here that maybe some of you are noting, who probably contributed to it, and that is the last decade, I think, Rich's discoveries regarding an enigmatic epithelial cell type called the Tuft cell, where he's really helping close in on a century-old mystery. And so with those multiple contributions, Rich has, of course, received numerous accolades. This is just a few of them, and I think we can be sure there'll be more to come. But I wanted to come back to this question because while Versa got it right about the amazing science, there's more. And I think high on that list is that he's been an outstanding mentor, and the list of trainee successes is incredible. And we're fortunate to have, I think, quite a few of them here today. And they were very happy to come back because of really that fantastic mentoring. Rich is also known for being the director of the Sandler Asthma Basic Research Center for more than 20 years now, and that's included recruiting some outstanding faculty and building bridges with physician scientists, such as in the Airway Clinical Research Center. And those bridges have helped advance asthma research at UCSF and well beyond. Rich is known for his teaching in the medical school and the graduate school. He's been doing that ever since he came here, and he's won many awards for his engaging style and for his scientific citizenship. And us immunologists have really just benefited enormously that Rich is always there at the immunology events, the journal clubs, the seminar series, these important training events. And that's not just that he's there, but his encyclopedic knowledge of biology and medicine, his incisive thinking, and his remarkable ability to see the big picture means that when he asks a question, which he usually does, it's illuminating, for all of us to understand the talk better, and really inspires us all to think more incisively and see the big picture. And I think a lot of that connects to Rich's deep curiosity about science and enthusiasm and excitement. It's always palpable when you stop him to talk about the science. And you can see a recent picture here that he's looking excited to think about the possibility of visualizing any cytokine. But that curiosity made me think about, it reminded me of a quote from Carl Sagan that maybe some of you know, that every kid starts out as a natural born scientist, and then we beat it out of them. A few trickle through the system with their wonder and enthusiasm for science intact. And I think Rich didn't just get through, but he sustained that enthusiasm and wonder throughout his career to the benefit of us all. And then Rich, we know, is for keeping fit. He's always looking trim. He's out there jogging a lot. And I think it's also connects back to how in the earlier days that Rich's team, the Locksley lab, would beat the Weiss lab in the softball matches regularly. But looking at his CV, it was interesting to note that his first invited review when he was a chief resident here was entitled Fuel Utilization in Marathons, Implications for Performance. And the intro to this review was written by Holly Smith, the then chair of medicine. And he noted that Rich did have some self-interest in writing on this topic because Rich was running sub 2-hour, 30-minute marathons at that time. But I would encourage anyone interested in this topic or in a marathon career in science to read the review. It's actually really well written about, and shows Rich's depths of understanding of physiology and metabolism even way back then. And then finally, we know of Rich as a devoted family man. And just one personal recollection is I remember when his girls were small, it wasn't that Rich was just going to the sporting events, but he was coaching them, and not just coaching them, but becoming head coach up to 2007, that he won the San Francisco Little League Softball Coach of the Year award. And he does tell me that that's the award he's most proud of. But I want to come back to finish to the amazing science and say just one more thing. And that there are a few people who changed the way we think about an entire field, and Rich is one of those people. So please join me in welcoming Rich to give the 67th Faculty Research Lecture.
Yeah, so how do I follow that? I'm so honored and humbled knowing the past of the people who stood up and gave this talk, and particularly do it in front of colleagues and many of the trainees who came back. I'm so thankful for that. I was talking to some before saying this is almost like a lab meeting, and yet I feel like I'm in an HHMI review. And it's a very, very humbling experience to sit up here. If you look through the list of people who've done that. So I'll try to interest you in more. There's a couple titles I want to make, a couple things in the title I want to bring out and get back to. First is, why is it a conundrum? And second, why the worms? And so I'm going to start with this poster that I show here that we used in the back. And what you see here is in the background, if I can find a pointer, this is a worm, and in the back, granuloma beneath in the lamina propria of the small intestine, and it has to go there because the egg is ingested, the larvae gets out, it burrows in, and it has to molt to get to the next stage, which is to go out into the outside, live on this nice bed of villi where it encircles and lives the rest of its life, making thousands of eggs. And this is its lifestyle, meaning it has to keep you alive, secreting eggs for its entire life, which is about 40 days in the mouse. So the question is, how does it do that? And one aspect of the epithelium you'll notice is that the stem, all of this is one cell deep. I'm trying to find the pointer here. All of this is one cell deep. And all of these cells come from the stem cells at the bottom in green here, protected by Parnassus cells. So they're all in green, lit up by LGR5, the stem cell marker. Here it is. You see that pointer? Yeah, okay. But you notice over the worm, LGR5 is extinguished, and the Parnassus cells have become untethered. They're going to the top where they go out. There's nothing wrong with the architecture. In fact, the villi are longer over the worm. So how is this epithelium maintaining itself in the absence of this stem cell marker for adult stem cells? And that was something we took on with Ophir's lab in a collaboration with a student in his lab, Ice Brandness, and Adam Savage, who was a postdoc in the lab at the time. And they punched out, Adam had figured out a way to punch out the granulomas and the normal pieces of gut, and then to figure out what these things expressed. And what turned out is that the granulomas over the gut were expressing antigens like SCA1 that are known to be expressed in the fetal gut and not in the adult gut. And indeed, in making organoids, these grew like fetal organoids, would grow as spheroids rather than on the top there, normal organoids that have little primitive villi that are being developed. So this suggested that the worm had induced a return to this fetal expression pattern to make gut. The way I think of it, not as a developmental biologist, but as a developing fetus, you just want to cover real estate. You don't need to make enterocytes and goblet cells and all the remarkable cells that we make in adulthood. All you need to do is cover territory and build a tube. So you build fast. And that's what these animals do. In fact, as you can see, all the genes that are highly expressed over this granuloma induced by the worm are these fetal genes. And Sarah Viragova, who still works in collaborative efforts between our lab and Ophir's lab, has gone on with all the research that's accumulated since then to show that this is a conserved mechanism by which tissues regenerate without causing inflammation. That is, when you're developing, you need to undergo a lot of cell turnover and a lot of building without causing inflammation. And that's what these worms do. This is a conserved pathway from salamanders through zebrafish, through humans. And the way they do this is kind of cleaning the slate, reactivating programs that are in place. My point in bringing this up is that these are impressive biologic agents because they've been selected by millions of years of evolution. And sticking them in to ask questions can tell a lot about how we ourselves are using normal pathways to create healthy organs. Now, I'm not the first person to think about this. Virchow was thinking about these things in the late 1800s, and he was amazed that trichinella could go into a muscle cell, dedifferentiate, build a vasculature around itself, and wait until you died and had your bones ripped off by a carnivore to complete its life cycle. So these things just wait it out. They're really evolutionary tuned to be transmitted, because worms, unlike these parasites, unlike bacteria and viruses that all of the offspring are immediately infectious, worms are not. They have to go out into the outer world, go through several molts, often a different host, like a snail, meaning they have to keep you feeling fit enough to walk down to the river, take a poop, allow that excrement to get to a snail, and go through several molts before it gets back into an infected person, into a human being. So it's a remarkable biology, and that's some of what we'll share today. Now, I'll go quickly through a little bit of what Jason said. I was born in Minneapolis, lived there a month. I was an Army brat. I grew up in all the usual places. Korean War, my dad was gone. We were staying with my mom's family, but I got a dog out of it. That was my first dog. Then jump school, got to do all the parachute training at Pentagon, Army War Barracks, all the deal. The first time I remember science really coming up at our dinner table all the time, and in schools in the education system, was Sputnik. So I'm old. And I remember going out on our block and looking up, and everybody said there's Sputnik, which I highly doubt. It's about the size of a basketball. But I think you could see the booster rocket that was in orbit with it. And I remember at that time there was tremendous interest in science. And we started talking all the time about, scientific things. I started going to science fairs, were popping up all over, building cloud chambers and mating flies and all kinds of stuff. And then we got deployed over to the Sixth Fleet in Naples. I was a baseball player. I had to play soccer. I was going to get bullied, to be perfectly honest. And Glia Azzurri, the other blue team, was in Serie B. They had been relegated. People know the Premier League. And they won the Coppa. So the Coppa Cup, Coppa Italia, is a big deal. And a Serie B team never does that. But that got them into Serie A. The whole city was ablaze. And I started playing soccer. That was way before everybody played soccer. You didn't have to be very good. And my dad had retired with the Vietnam War. The kids were getting very radicalized. He took up a whole different career, taught at the University of Maryland for years. And I got sent off to a boarding school in Connecticut, Kent School, and played soccer and played baseball. Went to Harvard, kept playing soccer and baseball. There was a lot of protests going on, and I was out playing sports. Why is this relevant? It's relevant because in my senior year, I captained the team that went to the NCAA finals. So the Final Four was in the Bay Area. So this is the first time I ever came out here, out to the West. And I said, my parents had a postcard. They kept on their refrigerator for years. It said, someday I'm going to live here, where I've been longer than any other place I've ever lived, given my background. So when I was at Harvard, I worked in labs over at the Peterson Brigham, isolating ATPases from cardiac tissue. I thought it was going to go into cardiovascular disease. The guy I worked for, Alan Stam, had gone to Rochester Medical School. He said, this is a good place to go. War was raging, seemed like a good place to be. So I went to Rochester Medical School, and I ran into Gordon Douglas, who was the chief of infectious disease. This is a very charismatic virologist. He went on to become chief of medicine at Cornell, and then the head of vaccine research at Merck for many, many years, and wrote the textbook in infectious disease, Essentials of Infectious Disease. And he just was so enamored by the idea of evolution in a body. He was a virologist. How can this happen? We're just two genomes competing, and it's all about transmission. Infectious disease isn't about antibiotics, as Dick Jacobs would say. It's about where did it come from, where did you get it, and where's it going? When you understand that, you see how it's so fascinating. And the other person was Bill Morgan, who was the acting chief of medicine. And when I'd finished my training, I said, I'm going to go back to Boston to the Peterson Brigham and the Mass General. And he said, I worked with a guy, Ollie Smith at the Mass General, and he took off and went to the West Coast. I think he's built a really good system out there. I think you should go out West and look at it. And I met Holly. I actually got pulled out of the line to go talk to Holly when I was interviewing here. And I knew there was not a chance I was going anywhere else. And Holly said, later showed me Bill Morgan's note. It said, "Dear Holly, take him. He'll be your chief resident." Now, I never know whether Holly just faked that note. But what it meant is that he wrote me into a chief residency. So I was stuck for 4 years doing this stuff in a very tumultuous time in San Francisco. So I started with being on in the ER with the Milk Moscone murders, the Jonestown massacre was right around then. Some people might remember the Chinatown Grand Dragon massacre, a lot of stuff happening, and ended in May of 1980 with the admission of the first patients with pneumocystis and Kaposi sarcoma. And so that's me over in the far thing. I think if you look sharply, I'll try to help you with a pointer. You can see a rather dour Joel Ernst sitting up there. If you're attentive, you'll notice a lot of other faculty members in that picture on the medical side of things. So at the time, I would go into Holly's office. We'd meet every Friday, and I'd say, Holly, I think this chief of medicine thing's a good deal. And he said, medicine's changing. You don't want to do this. You go get an education in infectious disease. I'll bring you back to be head of ID. It was a really man's world, for lack of a privileged world, for lack of a better word. And so I went up to Seattle and trained with Seymour Klebanoff, who was an MD, PhD, actually started out in endocrinology, who had identified the way thyroid hormone is iodinated by thyroid peroxidase. Seymour then went to train with Zanville Cohn at Rockefeller and Macrophage University back then, and got very interested in myeloperoxidase, which is another member of the heme peroxidase family, and determined how myeloperoxidase, which is most of the protein in neutrophil, killed by making hypochlorous acid, so it fixes the chlorine, and so all of these heme peroxidase fix these iodides, fix these halides. So it makes bleach, and through Fenton reaction kinetics, it makes radical oxygen to kill organisms with hydrogen peroxide in the oxidative burst. So he was very interested in eosinophil peroxidase, which was very cationic and stayed around would be discharged from eosinophils, and whether it killed parasites as a bystander, because it would absorb onto surfaces. And he knew that tissue macrophages had lost myeloperoxidase in their residency in tissue, and whether this would arm their relatively weak oxidative bursts and allow them to kill. And the target was brought by Chris Wilson, who was a new assistant professor from Stanford, who was working on toxoplasma from Jack Remington's lab, and been publishing in JEM about how toxoplasma really lowered the burst. So it was a good model. And so through a whole series of things involved my going up into a horse slaughterhouse, because it doesn't exist anymore north of Seattle, collecting tons of blood from horses and purifying enough eosinophil peroxidase to do experiments for a long time, I could show that if you got eosinophil peroxidase onto the surface of these cells, they would be killed. And this is one of those random things that happens in science for trainees. A reviewer said, you should be working with a protozoan that merges with a lysosome like Leishmania. I knew relatively nothing about it. But once I read everything about it, I knew I was going to work on it. So the Australians, a guy named Graham Mitchell, a very sharp guy, had done a lot of work in working out this problem, which is a foot up, foot down problem, which I really liked. This is a disease transmitted by sandflies, and the organisms infect macrophages. And by that time, right around 83, I guess it was Carl Nathan, Hank Murphy, people at Rockefeller were identifying macrophage activating factor in helper T cells that now had monoclonal antibodies that could find them, that were producing a cytokine, interferon gamma, that activated macrophages to kill. And it took no time at all for us to confirm that that's how the healer mice were working. They were secreting the interferon gamma. So then Holly called me up, said time to come back, and he brought back the chief of medicine from UW, replaced him, Dick Root, and I came down to take over infectious disease. And just at that time, Mossman and Kaufman down at DNAX, which is owned by Shearing Plow back in '86, had pulled a bunch of clone T cells out of the freezer and showed that they all either made interferon gamma and some other stuff, TNF, things like that, and they called them Th1 cells, or they made IL-4, this factor called B-cell stimulating factor back then, but they had the cDNAs. I knew right away that was what was going on. Drove down there. Ken Arai, I think, just gave me the CDA. I know Lewis was down there then. I mean, there was no protagonist. I think I signed as an official of UCSF and just took it back. I didn't think anything of it. And in no time at all, we had showed that this was in vitro, a very good in vitro example of what they could see in clone T cells. Now, and this has been picked up in textbooks, which rarely happens in your life. Here by Bill Paul, who was really the first person to isolate and study IL-4. And I think Tony may have been in his lab when a lot of this stuff was going on. So fast forward, these are those papers in macrophage. It was called Journal of Mouse Macrophages, JM. And it was a pretty straightforward northern blot. It either had interferon from the healer mice or IL-4 from the non-healer mice. But the really interesting insight that we had was we treated these mice with anti-IL-4 early, and they reversed their capacity, and the susceptible mice could generate these protective T cells, which was quite an undertaking that we should have patented back when you could patent natural molecules. But aside from that, this underlined that cytokines were the language of the immune system and that words matter. Now, the second thing that happened was Genome Project was starting, and Eddie Rubin, who was an MD PhD with Y.W. Kahn here at UCSF, went over to Livermore to start on human chromosome 5. Chromosome 5 is right where the cytokine locus is, where all these genes are gene duplicates. And the core cytokines we're going to talk about are 4, 5, and 13, all of which are targets for asthma and allergy confirmed in humans. So they were sequencing across there, and they were very concerned because, like everybody doing the human genome, they were hitting all this non-coding stuff. They hardly get any genes. And they were plowing along, but they were smart enough to realize that very long sequences were conserved by comparing along a whole raft of species, like chicken, all the way down to blowfish, to fugu. And so by comparing sequences, they were looking for non-coding sequences, at least 100 base pairs, in bold, 1 copy in the genome, and an asterisk conserved by 7 vertebrates from man to zebrafish. And the longest hit was right between IL-4 and IL-13, called CNS1. So we got together to start looking at this. They made a yak out of this interval H24 up there where they could go in and delete CNS1, and then you can put it into mouse cells. And of course, mouse cells don't make human interferon, but we could show that if you deleted in that middle line, if you deleted this unit, then the cells either made 4, 13, or 5. And then Marcus Moores in the lab deleted it out of the, once we got it onto a good background, deleted it out of the mouse system, and we saw the same thing. So cytokine words seem to be in sentences. And these things at the core of the type 2 locus are very important. Now, what's important about this biology is thinking about the disease it causes, which is quite different in developed and developing countries, and was the focus of something that was advanced as the hygiene hypothesis by David Strachan in 1989. And what was evident that in developed countries, more and more people were developing asthma, allergy, huge increases in prevalence from the late 60s to 2000. Most of those patients had allergy. Food allergy was beginning in the 1990s and going up such that now almost 1 in 10 kids have a food allergy. And in the generation since I went to school eating peanut butter sandwich almost every day, it was outlawed at my kids' school. So clearly this is environmental. Now it's the most allergy or atopic diseases are the most prevalent diseases of childhood in the US. They cost an astronomical amount of money. But the exact same type of immunity, which I'm calling type 2 immunity because the names of those cytokine loci in Th2 cells, which really drove the adoption of the use of type 2 immunity as allergy. In developing countries, parasite infections are associated with all of these same things: eosinophilia, high IgE, Th2 cells, all alternatively activated macrophages. It was coined by Simon Gordon to point out non-inflamed macrophages activated by IL-4/13 that were really good at taking up apoptotic corpses, for instance, in a very non-inflammatory way. So all of these people, billions of people, intestinal roundworms, these live in our small intestine, one epithelial cell away from the absolute organ that we need to get all the nutrients for our body. And all these other organisms, and they certainly cause disease, so I don't want to mislead you, but almost all infection is asymptomatic in these many, many people. And the morbidity and mortality shows up after the reproductive years. So from the point of view of the worm has only got to get all of its eggs out into the environment. From the point of view of the human, they only have to keep you, or whoever the host is, they just have to keep you healthy enough to get you to not select a gene that selects against them. So appreciating that, and this is in wild vertebrates in the most extreme places on the planet. Penguins, where you think of their selection against this, would have long ago disappeared. Penguins have the same number of worms per animal as we would in the right setting where we're exposed. So these are great reagents. So when you look at the history of allergy and asthma, this is the conundrum. So if people have allergy, everybody says, oh, what are you allergic to? You're allergic to cats, or you're allergic to shrimp, or you're allergic to peanuts, which is by definition adaptive immunity. These are antibodies like IgE that see these things generally, or T cells that react to them. And the recognition of allergy is littered with horrible things. So Ehrlich really identified all these cells with special stains, and Richer got a Nobel Prize for anaphylaxis. Ehrlich got a Nobel Prize with Metchnikov in 1908. And then Prosnitz and Kussner did this incredible reaction where Kussner had awful fish allergy, and his Prosnitz got serum, injected it in skin, and then ate some fish and lit up with hives where he'd injected the serum. But of course, that toll was transferred. I have no idea how he died, but I hate to think about it. This led to the elucidation of IgE as critical in this whole thing. And then Mossman and Kaufman, I've told you about these T cells named for their type of cytokine. Type 1 cells, type 17 cells, it should be type 3, and type 2 cells that orchestrate different aspects of biology in the same organized way that these core cytokines organize in Th2 cells, and then regulatory T cells that regulate the adaptive phase. But of course, the genomic era, we were finding all these things. You could find all of these proteins and mRNAs in human samples. We began to be able to manipulate these in the mouse, knock them all out, and show you couldn't get allergy anymore. And now these are documented targets in human disease. All of these are therapeutics that are used in human asthma. And they're not perfect, but they clearly work extremely well. And probably the best, and I have no axe to grind here, is the anti-IL-4 receptor that's shared between the type 2 IL-4 primary 13 receptor in all tissues and the type 1 receptor that's on lymphoid cells, as was the way follicular T cells talk to B cells in germinal centers to shift to the heavy chain used to IgE. So we and a couple other 3 different continents discovered ILC2s at the same time, and around the same time in 2010, and that raised a quandary. What does this have to do with allergy? And I'm just putting on the top this hygiene hypothesis. Hay fever came when houses got more contained and you started to build up all kinds of exposures indoors. Asthma started increasing with pollution, but pollution was clearing out by the 1990s as the incidence was going up. It clearly was not related to pollution. There's the famous merging of East and West Germany. The incidence of asthma went strikingly up in West Germany, not in East Germany. It was lower in East Germany to start with. And increases in metabolic disease and increases in food allergy to almost 1 in 2 kids. So was it possible that innate cells were contributing to this? So I was thinking about this. This isn't just kind of a thought piece that I wrote with Doug Fearon in '96, but we lacked the tools to really address it directly. But now that we had these cells, we could do this. And what we now know, I'm just going to fast forward to a whole bunch of data, but a lot accrued here through people like Steve Van Dyken worked on this, and Roberto, a lot of people in the lab. But what we knew with help of people like Jimmy E and Alex Marzen was that these cells shared with Th2 cells that obviously have this receptor, so they can see antigen, where ILCs only are seeing other tissues. The ILC2s are poorly named. I've argued for IH2 on the nomenclature committee, but got nowhere. So it's innate lymphoid cell group 2. And so, yeah, it gets confusing when you say ILC2 cells, but that's the way it is. They have the same transcriptional landscape, the same chromatin accessibility, overlapping super-enhancer landscape. So large areas of genomic expression are open. And terminal tissue licensing, really shown first by Steve, I believe, in the lab, and Jesse, occurs through interaction with certain proteins called alarmins from different families. 33 is in the IL-1 family, TSLP is a 4-alpha helix bundle protein, 25 IL-17 receptor protein. And this worked because they put up these receptors. And this happened in development, independent. You could show this in germ-free mice. So this is developmentally regulated, tissue-specific ways, but they required licensing, as others have called confirmatory signals, to activate these signals and then see these alarmins in tissue and get licensed. So we already knew something was afoot in old experiments. I think Jane was even involved in some of these experiments, Jane Grogan, when she was here. And that was using schistosomes when Jim McCarroll was here as head of pathology. And you can see on the far left here that these granulomas are well formed. I can't do this without looking at it. They're well formed, but if you knock out IL-4, IL-13, the animals bleed out and die. They don't form granule, everything's gone. But by then we had tools where we could knock Flox the locust. David Veringer did great kind of old style back recombinering to get the LOX-P sites in. And we could knock 413 out, and none of the 413 that was engineered, that was doing this, was coming from adaptive T cells. So that was insightful. Then Davina Wu, I think this is a first paper on the in the lab that Ari worked on to finish up. I showed that the signature of type 2 cytokines, that is alternatively activated macrophages and eosinophils, were all over healthy fat. And that as fat became inflamed, you made obese mice, these disappeared, and you got inflamed macrophages that contributed to tissue resistance. And this explained in follow-up studies, the experiments we did with the Chawla lab, with a lot of different mechanisms in other labs as well, contributed to a pathway by which ILC2s maintain this healthy phenotype, and explain the finding that you can infect an animal with a helmet and do standard metabolic testing, like insulin glucose tolerance tests or insulin or glucose sensitivity or insulin sensitivity, to show there was amazing effects on metabolism that were contrary to what you saw in an inflammatory infection. And another one was started by Tiffany Reese, who had found that chitin, which is kind of molecular bones of a lot of allergens, would induce this type 2 response. And Steve Van Dyke had carried this forward to find out what genes were downstream of STAT6 signaling, which is the transducer of the IL-4/13 receptor. And the highest upregulated gene in the lung was chitinase. And humans don't make N-acetylglucosamine, this complex polysaccharide. It's insoluble, indigestible. But Steve could show that it was induced in club cells in the lung. And then if he knocked it out, these animals all died. And they died because of chitin that was in the bedding. And they accumulated in their lung lots of undigested chitin that drove this alarmin perturbation state and stress in epithelial cells that drove the ILCs to activate through all the pathways contributed, and then started generating additional alarmins that activated Th17 cells and gamma delta cells to make 17 that brought in neutrophils and led to fibrosis in death. And if Steve knocked out the IL-4 pathway, these animals fell over dead even earlier. So chitin is everywhere. It's all over this room. I guarantee you're all breathing it in as we speak. This is the bones of invertebrates. You can find it in dust with chitin binding domains, but you can find it all over the world. And my favorite site is at these vents, volcanic vents where plasma the magma comes up between the tectonic plates. And deep under 2 miles down, no photons, tons of pressure. These vents are gassing hydrogen sulfide. And the life form that springs up around these volcanic vents is tube worms. These are annelid worms that live in a chitinous home where they get shelter. They attach this chitin onto the top of this incredible things with archaea commensals that are metabolizing a sulfide-based chemistry to carbon metabolites that can be utilized by the worms. These are some of the longest lived and fastest growing creatures on the planet. And for Jen's sake, they're almost my favorite thing beside tardigrades. So these are incredibly interesting organisms, but just show you how diverse this thing is. Some people think life may have started at it, but back to the topic at hand. Steve went on to show that this enzyme was secreted by club cells. This is a secreted enzyme into bronchoalveolar lavage with help of Peter Walters, who takes care of a lot of people with interstitial lung disease and pulmonary fibrosis here. And it wasn't missing, and it wasn't the other chitinase, which is made largely in lysosomes of Kupffer cells and resident macrophages and other tissues. And these patients accumulate large amounts of chitin in their bronchoalveolar fluid. So it's not keeping up with clearance. So I just tell you a brief aside with a fun interaction we had with Jamie Fraser's lab. Anybody who's worked with Jamie knows it's always a fun interaction. And Jamie and Steve, we continue to all kind of work together on this. Two students of him, Ben Barad and Robbie Diaz, worked very hard to mutagenize chitinase and then pass it in a droplet system through a fluorescent N-acetyl glucosamine fluorescent dimer that we could split and quantitatively pick better chitinase. Humans don't have very good chitinase. You get away from the ground, it's even worse chitinase than mouse has. And then reiteratively express better chitinase. And Steve, has really, you can use Michaelis-Menten kinetics, select these better chitinases, and then take them into mice that are knocked out for chitinase and show that you can improve response to things like fibrotic lung disease induced by bleomycin. And so the concept here is a nebulized optimized chitinase, kind of like people use DNase or mucolytics to treat inflammatory diseases, either to get a handle on better therapeutics, put off transplantation, or waiting for better therapies, like has occurred with CF. So now that we could see these cells, a big surprise came from studies led by Jesse Nussbaum, who's here, I'm happy to say. And that was that we had to get out of all those tissues I had learned about at the bottom as an immunologist and focus in all the tissues of the bodies. So that's where ILC2s were, as shown by this IL-5 promoter, this odd cytokine that supports the growth of eosinophils and B1 B cells that contribute to IgA in the gut and to germline sequences that are selected early in development. So these cells are showing up in large numbers in all the internal organs, and particularly barrier organs like skin and small intestine and uterus. So this forced us to begin to wonder, is this real? And it became very apparent that that was so. That is, if you took away the adaptive immune system by crossing these onto rag mice, then serum IL-5 is sustained. But if you made one allele, it went to half. And if you took away a lymphoid cells, serum IL-5 and eosinophils went away. So innate cells in the tissue were making serum IL-5. So that kind of interesting. It had been said that eosinophils for a long time have felt to be circadian, and that the circadian rhythm underlies the fact that asthma patients always come at night to the ER when eosinophils are high. Whereas mice are reversed. And so we could figure out by judicious use of making these better reporters how this system worked in tissues. And under basal states and in SPF conditions, these mice in my pointer. These mice really were just making IL-5. But as soon as we perturbed the tissue, they made both 5 and 13 from the most activated cells. And that, we knew, 13 induced eotaxins from various stromal cells, fibroblasts in close proximity to ILC2s that Ari works on, and VCAM, the tethering molecule on the endothelium that's required for eosinophils to come to the tissue, that's lowering the blood eosinophil count. And you got alternatively activated macrophages. So they changed the tissue environment. So we figured we could sort out how the circadian rhythm was going, first by looking at isolating away from the metabolic rhythm. And Jesse did that by adapting mice to day and night feeding. And once they were adapted, he could look in fasted mice. And in fasted mice, you could see these cells aren't very activated. And when he gavaged food, 4 hours later, the innate ILC2s lit up. And that was surprising. And it didn't matter whether we reversed the light cycles or not. It had nothing to do with circadian rhythm. But we were very interested in this. And we were beginning to learn about receptors on their surface by now. One that was very prominent was the VIP receptor. It was well known that VIP was very involved in motility post-eating. It was induced in enteric neurons. And Jesse could show that it interacted with BPAC2, which is very highly expressed. VIP is the secretin glucagon family of proteins that we'll come back to. So with that, I'll fast forward to some recent studies looking at what might be driving 13 with a alarmin, TSLP, from an old mouse we generated some time ago, but Chong in the lab has really done a great job dissecting that. And that's showing here that TSLP is expressed in these subepithelial fibroblasts more proximally in the proximal bowel, in the distal villus. But in the distal bowel, most of the pericryptal fibroblasts down at the bottom and up at the top up here. And by basically sequencing these cells, we can very quickly tell what these cells are. We can tell by where they were located. These are what are specialized fibroblasts called telocytes and trophocytes that regulate the Wnt-BMP gradients that determine differentiation of epithelium. With the trophocytes down here, keeping cells stem-like, and the telocytes driving the Wnt and non-classical Wnt and BMP gradients that allow cells to differentiate. And she could show that all the telocyte populations did that. So with that in mind, we began to wonder how nutrients are being detected, which are seen by specialized endocrine cells called L cells that release gut hormones. And these fibroblasts, the telocytes, sit exactly below the basal membrane in contact with the siphon from these endocrine cells. And by sequencing, we could line up all of the different the telocyte and trophocyte populations. And it was clear they all express the GLP-2 receptor, GLP-2. I'll call GLP-2. And that all of these expressed these telocyte transcription factor FOXL1. And going into human databases, we could pull out the exact same cells. This is from the Haber lab, just showing TSLP and GLP-2 are expressed in the same cell. So it's GLP-2. So GLP-2 comes from the processing of a pro-glucagon gene that most people are aware of as made in alpha cells of the pancreas, processed by proconvertase 2, and then important in making glucagon, which is important in protecting us from hypoglycemia. Some people may not know that glucagon is proglucagon is very highly expressed in the L cells in the pancreas and in neurons, where it's processed by a different protease, proconvertase 1 and 3, and that generates GLP-1 and GLP-2. Now, you'd have to be sitting in a cave for we did not know what GLP-1 is. It just won Alaska and won the Breakthrough Prize for amazing effects in type 2 diabetes and on obesity. So there's incredible effects not only on health, metabolic health, but on health in many systems, as you're probably aware, cardiovascular system, kidney. It's being evaluated in neurodegenerative disease, in post-transplant disease. So it's quite remarkable. It's been well reviewed. What people may not know is that GLP-2 is made equimolar to GLP-1 and is also a drug. that has been stabilized and is used for short bowel syndrome. So neonates are born with short bowels that don't fully differentiate, and it's used for parenteral nutrition because you atrophy your bowel when you don't have luminal content, and growth back requires GLP-2. So these cells make GLP-2, and Chong was interested, and since these fibroblasts have the GLP-2 receptor, did that release the TSLP that they also had? And in fact, if she did this feeding routine under the same kind of system, this activation of 13 ILC2s was absent in TSLP receptor mice, or mice that have receptor cleaved from innate lymphoid cells, or mice that were deficient in GLP-2 from the Drucker lab. And if we use the GLP-2 drug that's used in human, it's conserved. If we did the GLP-2 drug, you didn't see this increase in the numbers of 13 activated cells. And you can flox TSLP out of fibroblasts with two different CREs, PDGRF alpha and a nestin CRE, and show that that was required. So it looked like the pathway, the Drucker lab, I should point out, had shown out that neuroreceptors for GLP-2 were driving the release of VIP from enteroendocrine cell populations. So it looks like innate lymphoid cells are tied in very tightly to a processing of luminal content. So the other thing I'm going to cover is, hopefully, is this understanding that tissues also have memory. And since I've shown you that ILCs are deeply involved in basal metabolic pathways, the question is whether these affect the memory status of a cell. So people have noticed in this nice review from Sharita Nook and Elaine Fugue that long-lived cells develop epigenetic changes after perturbation, and those can be retained at various cells in the organ. But what the hierarchical organization of this memory is not clear. But what is known is that there's a lot of difference between physiologic responses and antipathogen responses as compared to pathologic responses that lead to inflammatory diseases down the line. So the question is, could we identify host pathways split away from worm pathways and identify some of those things I showed you that parasites do? That is, they seem to help your metabolism, and they seem to do a lot of things that are counterproductive for things we think of as infectious disease. So we're using this as an evolutionary probe of this. So we went to the intestine, the single cell a barrier tissue, and we knew from early studies that ILC2s express very high levels of the IL-25 receptor, the ligand binding domain, IL-17RB. Protein expression on lamina-propria ILC2s is very high. And we could show that in the gut, if you perturb the gut, now with Nipostrongis, the ILC2s are already activated. And they start proliferating and activate well before Th2 cells appear, which is around day 7 or 9. And at that time, the worm is being expelled in the system. And if you knock out 25, you can see that this totally affects the clearance of the worm just by taking away this innate source of 25. You might not be aware that 25 is a 17 family member, hence the receptor designation. It's a very ancient cytokine that's in marine invertebrates like sponges and tunicates that are basically worms. And worms like C. elegans have IL-25 genes. If you disrupt these, you disrupt the food sensing pathway. So this is a very deeply ingrained pathway in food sensing and the environment. And one of the this is another foot up, foot down experiment done in the lab, is when we labeled ILC2 in the work that Jakob did in the lab, and got this spectacular response with all of these cells that got magnified. And this is a mind-boggling experiment because these turned out to be tough cells, this chemosensory sensing cell. And so I'll just kind of highlight some of the things we overlook about that series of studies from his work and from work of others who were contemporaneous with that, that linked epithelial sensing with innate immune cells, established methods to grow tough cells. These things cannot be grown in vitro until 13 was identified as a driver, and revealed this dynamic circuit that could link sensing to weep and sweep because it changed the secretory balance of the epithelium to goblet cells and tough cells themselves. And it's dramatically accelerated. If you just look at PubMed, the numbers of studies of tough cells have gone up dramatically. And just an example of the tremendous numbers of goblet cells that come up very quickly after nipostrongos. It's entirely due to IL-25 made in the epithelium using the villain Cree. So these are solitary human sensory cells. What was interesting is they had very stereotyped outputs that were unusual for an epithelial cell, a neurotransmitter, acetylcholine, this IL-25, icosanoids. Christoph's done some nice work looking at how the signaling goes through IP3 receptor 3 to cause the release of IL-25, and how 17RB itself on tough cells can autoregulate this system. They had been described for over 50 years in vertebrates, really not knowing what they did. And now many, many ligands have been found. They've been shown to zonate by the Clevers lab, recently by IL-13 using the system we developed. And they all seem to make this very canonical pathway output, of which 3 of those receptors are expressed at rest in ILC2s. So another important experiment was again to find, this is Christoph when he gets back from surfing in the morning at Ocean Beach. And in this way, by knocking out 820, that deubiquinates the 17RB chain, as Sarah Gaffin had shown, and regulates the IL-25 response to make it not keep going in this feed-forward loop. We could develop mice that were stuck. And the important thing about this experiment was these mice were fine. They had no allergic pathology. They had a lot of remodeling with long guts, epithelial secretory cell bias. They had decreased visceral adipose, improved glucose tolerance, and they had concomitant immunity. They explained how other pathogens like other worms couldn't get in. The physical place that they have to undergo molting has been altered. And now that interrupts the development. From a worm's point of view, being anthropomorphic about it, you kind of keep the host to yourself at the point that you're in a reproductive mode. And so that really means you got to keep people healthy, because you got to get them to uninfected hosts. And all this was deleted if Christoph knocked out IL-25. So that's set the stage for the last story I'm going to tell you, which was summarized in a cartoon. But Victor Cortes spent a long time optimizing, but he optimized a regimen where he could condition these mice and then wait a long time. Plus 50 means that Victor's human. He didn't come in and take down every mice on exactly the same day. So we characterized mice between sometime plus 50 and sometimes plus 80. The mean is sometime around 73 days, to be short about it, but some mice have gone out a year and a half. So be that as it may, we could cut the bowel up and start asking what is held onto by IL-25 alone, this host response to this intestinal perturbation. And the lengthening is held on for 180 days after stopping. They've seen no IL-25. And if we look at the epithelium, it's greatly altered. That is, all of these are IL-13-induced genes. I won't go through what a lot of them are, but we could confirm many of these are expressed at the protein level, like S-dermins and CD55 that protect cells from complement. These tough cells were highly increased, but in the normal gradient, they were just increased compared to the normal proximal to distal increase. They had the same canonical signature, but interestingly, they had downregulated IL-25 itself. See how that's kind of interesting. And they increased the goblet cells. Over here, you can see with MUC2 staining. And all of these also express a number of IL-2 genes. I know Tiffany recognizes SBRR that she worked on with Laura Hooper in her own lab. So they had all this epithelial cell response to IL-13 that persisted. And of course, the ILCs and the Th2 cells were greatly increased in the gut. But surprisingly, the rest of the lymphoid populations are pretty unchanged, including Tregs. And that's quantitated here. And the amazing thing is that when using these reporters, Victor went in to take a look at what was going on. These cells remained activated. That is, they were cranking out within the limits of the reporter IL-13 and IL-5 for very long periods of time. And from the phenotype, these cells went all across the bowel like this. We'll talk about the phenotype in a minute. So here these cells are. You can see them easily. They're all up and down the villus, right next to that subepithelial space. And they have a phenotype, and they had a low proliferative rate that's consistent with memory effector cells that have been seen in a lot of lymphoid population. Really, super CAR T cells have this phenotype that we'll get into. That is, they're CD25 low. They downregulate kind of stem-like genes like Thy1 and SCA1. They don't proliferate much. They're KLRG high, activated, and they express these cytokines, and they're tissue resident. A lot of TGF beta receptors, a whole slew of TGF beta receptors. So when Victor did ATAC-seq on these cells, it was clear they had lots of changes, whereas the LGR5 crypt epithelial cells had insignificant changes that made no sense when we look at them, including by ATAC of organoids. And they were in the usual suspect regions like promoters, intergenic regions that we were interested in, and distal intergenic regions. And if we pulled up the 13, like just the Th2 locus, there were interesting changes all over the genome. So we could see interesting changes in things that, remember, all these cells express this anyway. It's an open locus. But you could see interesting things shown in the pink here that were hit, including the IL-13 promoter, HSA3, that was something I think Mark Ansel worked on in the Rao lab for a long time as an enhancer in the IL-4 gene. The silencer at HSS4 and the enhancer at the CNS1 both were altered, but not significantly so. So there were a lot of changes that explained a lot of things that we saw about these cells. And the telling point was that these cells, here with Sarah Varagova's help, grew in epithelioids identically, whether we made them from proximal, middle, or distal gut, from mice that had gotten 25 or not. If the memory was in the stem cells, we would have expected to see a lot of tough cells. And they had no different sensitivity to IL-13. We could induce tough cells. And furthermore, they developed crisscross experiments where they could make epithelioids from either control or 25, all age match treated the same way. Neither one made more tough cells, and only if they were incubated with ILC2s from the IL-25 treated mice did they develop this tough cell phenotype, and that phenotype could be completely abolished by incubating with anti-13 antibodies. So anti-13 and 4. So this indicated they had just reproduced Jakob's feed-forward loop in vitro. So in single cell sequencing these mice, they clearly moved to a different space. And I don't want to say they're all exactly the same cells, about 5, 6 interesting clusters here, but all of those clusters share the core components I'm going to talk to you about, with expression of some very interesting genes like ZEB2 that is involved in a lot of memory effector cell development in many lymphoid populations, from NKTs to NKs, CD8 memory effectors, CD4 memory effectors, TOX. That gene that if it goes one bump further correlates with exhaustion is highly expressed. And there's downregulation of a lot of things that set stem-like precursor phenotypes, but no loss of the lineage specific genes. I'm just showing GATA3. They express all of the alarmin receptors. That's what these are here. And some other things are really unchanged, but they open up the Th2 locus. But interestingly, GM-CSF, which you verified at the protein level, is completely extinguished. This is an inflammatory cytokine right next to IL-5 in the genome. And these cells have this phenotype as established by FACS. You can see where they move, but a non-stem-like. Now, it's not to say there aren't precursor stem cells in there. And now we're fast forwarding in the field, but it's known that the precursors get laid down through life in the tissues. And your Gastager has really pioneered these cells. But if we were sorting these cells based on activation marker, we would never get them. But there's definitely precursors in there. I don't want to mislead you, but the vast majority of ILC2s driving this phenotype are resident memory long-lived effectors. The intriguing part of this is these are natural populations. If IL-25 is knocked out, you see that there are normal cells already there in this space in wild-type mice. And if we treat the mice of 25, these cells never retreat. That is, you don't need 25 in development, and you don't need it once you achieve the state. This is a true Waddington type of gutter. And they have all of the exact same populations that we see in wild-type mice. Secondly, we see them in helminth infection, but they're induced in much smaller numbers in wild-type infection, and helminths begin to regulate this, as has been shown by others. And of course, we've cut the helminths out of this purposely for that very reason. And lastly, they're present in humans. This is a study from Jenny Mossberg, and I've highlighted there in green many that these things show, but things like ZEB2, ILC2 is in the middle here, ZEB2, HLF, which we see, which links into circadian rhythms, IL-13, all of the defining criteria, KLRG high, all TCF7, LEF7 low, which correlates with a more stem-like compartment. So the things you see in the tissue are these memory effector cells. What was really striking is that when they were crossed into mice that lacked all of the alarmins, these mice still could be induced, maintain their effector potential, and escaped alarmin. Now, if you take them out and activate an alarmin, they activate fine. And the way I think about this is the barrier has actually been fortified in a way that by this constant output of 13 and 5, that things that perturb take longer to accrue. And so they have a much higher threshold to be activated by alarmins. It may say something about whether alarmins are going to make a really good target, certain alarmins. And as best we can tell, all of these alarmin knockout mice recapitulate the entire phenotype. And this just shows that alarmin deficient mice don't get rid of any worms. So on the left here, 150 worms is all the worms we get back with our inoculum that mature in the gut. With just in these treated mice, just with these innate cells into a wild-type animal, you can recapitulate this protection. So there's these truly are natural populations that have this amazing long-lived phenotype. And they're required to drive the phenotype. So if you delete them by inducing DT, the tough cells fall almost immediately. Speaking this fact, they're constantly driving this epithelial escalator. And if you transfer them in Nippus strongylus, they protect mice that are lymphocyte deficient. So that's an important experiment because there's no niche for lymphocytes. So you might argue that, well, the 13s aren't making a niche and that's where they're required to sit and do their thing. They're being activated in situ by different inputs. If I was guessing, I would guess it's rewired neurons that's being shown in experiments by Charles Zucker and others, and a lot of interest in neuroimmunology field. And there's no obvious contribution to microbiota, which we know changes in these animals with all the mucus out in the GI tract. But if we do it in germ-free mice, the animals develop the phenotype normally. All these cells develop normally in germ-free mice. If we co-house or don't co-house the mice, it doesn't matter. If we transfer cecal flora from mice to germ-free mice, we don't induce the phenotype. And now I'll close with kind of my where do we go in this space? The truly interesting thing is these cells migrate and show up in borders of distal organs. So here they're in colon, they're in lung, they're in adipose. They have the same phenotype. You can see them. They're marked with intestinal imprinted signs like alpha-4 beta-7, CCR9. And it suggests that there's an outward movement of cells from the gut in the normal setting that we miss in SPF mice. And this has ramifications for survival in the wild. These animals are more resistant to salmonella. They're more resistant in experiments of Mike Diamond and Steve at WashU St. Louis. They're more resistant to SARS-CoV-2. And at the right doses, these animals are protected from death. They have good metabolic changes. And when we try to purposely give them food allergy, that is by sensitizing the ova, and you'd hate to get mice, you could wave a peanut over and they'd fall over dead. As best we can tell, they're no more sensitive to allergy than another mice. Again, for reasons we can talk about. I think it's fortification barrier. So what I've tried to convince you is that these innate cells that get laid down in development are very integrated into the regeneration and homeostasis of tissues. And we've missed this by working in SPF mice. And I've tried to outline the steps, the basal biologic steps in the nutrient pathway that helminths that presume to live in this space that is so critical to us, and we simply ignore it and assist their reproductive lifestyle. But helminths hijack this system. They cut in at the tough cell level, another protus in the mouse, and activate the tough cell and activate this internal circuit from outside in, rather than interrupting the normal food circuit that goes on normally in these mice. And IL-25 seems to mimic this host resilience while avoiding a lot of the pathology that is induced by helminths. So it's kind of outlined schematically here in a slide that Victor dummied up, but we didn't see this coming, but I will tell you in a slide I left out just in the interest of time, Roberto and Miliana, a postdoc in the lab, have shown that Demodex parasites in the skin elicit activation of very easy to see skin imprinted ILC2s that in colonized wild type mice with low levels of Demodex, these cells go to the lung. So normally the lung is being educated post-birth outside in by innate lymphoid cells educated at the barrier by pathobionts. So that's kind of summarized here, this idea that pathobionts, we know early asthma allergy is basically a disease of barriers where things get in inappropriately. And this state is probably involved in maintaining optimal barrier status as we move from the fetus, where barriers don't matter, to borders inside and outside the mouse once you're born. And I think learning about this therapeutic option offers a lot of ways to think about developing biomarkers to assess where people are in their state and how we might make them go there using molecular mechanisms and defined ways rather than complex parasites evolved over millions of years. So with that, I will stop. Thank all of you for coming. What an honor. I've tried to name everybody going through this whole list. I can hardly get through this. Great people. Could have done any without all of them. I'm proud of all the people I've co-published with here at UCSF. Pretty amazing list of people. Never Art Weiss. I sat next to Art Weiss, who I admire more than anybody else in the world as a scientist. And we would talk science all day, but I think he has the same kind of excited thing of doing what he's doing. And gosh, what a lab partner. And of course, these two guys have been with me for ages. Wong running all the flow cytometry and Onger developed all this genetic manipulation. It's been such a boon and a help. No complaints. Working with many, many labs all over the hospital. And of course, people in the Saber Center, and I haven't been able to talk about all my ID colleagues. I see some of them here. I'm so grateful for them coming. I consider Joe Guglielmo became chief of pharmacy. I consider him an ID colleague because in the old days, he would round with us on ID and coach us on all the pharmaceuticals. And of course, my family, hard to get through. Kari worked here for 37 years, right, as a rheumatologist, dermatologist, dermatopathologist, and my kids saw the light, I think, do other things. But what great kids, what a great family. And my parents are buried in Washington at Arlington Cemetery, so I can go back and see them when I go back to Hughes and to HHMI. And at home, Kari and I have these guys who to keep us company. Thank you all so much. Sorry.
So
All talked out.
On behalf of the Academic Senate Committee on
Appropriate?
Research,
It's appropriate.
we'd love to present you with this certificate for your achievements in basic science research.
Wow. Wow. This will replace the coach of San Francisco Little League softball. Or we do questions.
So we'll take a question from here in the room, and then we'll alternate on Zoom. So is there somebody here who would like to ask questions?
Yeah, sure. Repeated injections of IL-25. You say that simulates a helmet infection or some other? The repeated injections of IL-25. Is the goal that to simulate a helmet infection, or is there some other thing that's mimicking?
Other things hit that reaction. Certain protists in the gut do the same thing. Do I have to repeat the question? Do I have to repeat the question for Zoom? Yeah. Oh, okay. So as long as I have the mic. Yeah, you're mimicking the host response to perturbation through the small intestine, but leaving out, of course, all the eggs, the wayward migration, all the other active products, as many people have shown in helminths. And that was the idea, was how could we segregate away the protective things that we know helminths induce, but not get into all the side effects. So yeah, it's helminths, but a number of things, including some bacteria, go through this pathway, as Michael Howitt and others have shown. So it's to really emphasize that host response, but maximize it. But I think in the developing world, we probably did maximize that border very early in life, because your mom would be infected with all those things and pass it directly to you very early in life and drive this. But we don't do that anymore.
I am Dick Jacobs. I was in the Division of Infectious Diseases when Rich was the Chief of Infectious Diseases. And everybody in this room knows after this lecture what an incredible scientist he is. I don't want anybody to leave the room thinking that Rich is unidimensional. He was a phenomenal clinician. I would say that hands down, without any question, he was absolutely the best at bringing the bench to the bedside. And he stimulated not only people in the laboratory, but stimulated clinicians to think about things in an in-depth kind of way about the etiology and pathophysiology of disease. I have one question, and my question is, can you imagine how intimidating it was as a resident to have Rich Locksley as your intern and try to teach him something?
Thanks, Dick.
Liz, is there a Zoom question?
Hi, we actually don't have any questions on the Zoom right now. If anybody on the Zoom, otherwise I'll go back to the room.
Thank you.
Great, fantastic lecture. Going back to hygiene hypothesis, like, do you guys know whether these memory effector ILC2s, when they really come up? And then I'm also as you're exposed to everything, and then I'm also wondering what happens in aging. As these cells go all around, do they expand or crash or?
Yeah, great question. You may have noticed that in the very aged mice, so I think 180 days, but Victor can correct me if I'm wrong, the appearance of these cells in tissue is going up. If you looked at the small intestine length, it lengthens with age, and our vivarium is not that clean. And once you get T. Muris in there driving this thing, slowly, these cells accumulate over time. But I don't think we hit it the way you would hit it in evolution, when we were picking up worms directly and many other things that drove this pathway to fortify barriers, both internal and external, very early on. So when do they come up? Victor did a fairly careful study, 1 week, 2 weeks, 3 weeks, and at 2 weeks and half the dose, I think one mouse reverted. And unfortunately, I, in my wisdom, said double the time, double the dose. It's got to work every time. And he did that, but it costs a fortune. To do this was ridiculous. So somewhere around 2 weeks, you begin to hit the sweet spot when you can drive what I believe. And in humans, of course, you see in aged human this barrier. But is it as good as it could be? Because if the barrier develops late, you may already have begun to develop a dysregulated adaptive response. Right? I think this develops very early in postnatal life.
Fantastic lecture, Rich.
What a great student. She always has a question. I came here.
I have to ask, what are your thoughts on these memory ILC2s migrating compared to other ILC2s, and also about the fact that they're offering protection in other sites in other disease model as well? And do you think that's also acting through epithelium there?
Yeah, so thanks for bringing that up. So we didn't discover migration of ILC2s. This is really discovered by Bill Paul and Wang in his lab. And a remarkable finding at the time, and confirmed by studies by Ron Germain, really showing how they egress and go through thoracic duct and get into the get to the lung and can adapt to a site of perturbation. So of course the lung is damaged, and so more cells accumulate there. So there's a give and a take. When you drive cells out of a niche, they have to have a place to go and outcompete cells that live somewhere else. And a little inflammation can salt the niche, we think. I'm hand waving a bit. Salt the niche, but me and Roberto are really looking at this carefully to look at how this niche controls the number of adaptive T cells that take up in this niche, and then may bring in a little bit of mischief, just given the promiscuity of the TCR.
Questions on Zoom?
Nope, still nothing on the Zoom.
Any more questions? Oh.
Great.
Very inspiring presentation. I was actually going to follow up on the last thought of why do you think we evolved to have both Th2 and ILC2 cells? They seem to be predominantly responding to cytokines in the tissue rather than responding to specific antigens. So why do you think we evolved to have both?
So in the case of IL-25, or
it's one of the signals that they both respond to, yeah.
So, say it one more time, not quite.
So why did we evolve to have both Th2 and ILC2 cells? They seem to be doing the same thing, basically.
Yes, so why do you have them? There's no question, don't get me wrong here. You need adaptive T cells. Yeah. If you give this helmet to a rag mouse, it will have a lot of trouble getting rid of worms. You can show the innate cells expand like crazy and release their type 2 cytokine, but they don't expel worms. And IL-2 from the incoming Th2 cells is part of that growth regulation, and part of it is cell-cell interaction, depending on labs that you read, and part of it is some interesting other molecules that may be coming from neurons. But there's no question that the adaptive response comes in and amplifies expansion of innate lymphoid cells. They're very rooted at the hip, if you will. And they're bringing the TCR in as a hedge of bet, that if this same peptide comes in again, we're going to make sure, we have another way to see this perturbation. But it has to get to a big enough place that they're ejected. And it's probably right around the threshold when dendritic cells are ejected, I would guess, or that migrate. But formally, we don't know.
Rich, does IgA play any role in helminth infection? And if so, do ILCs have FC receptors for IgA?
They don't have FC receptors, to my knowledge. There's people here now who probably could tell me that. IgA has been brought up over and over again in terms of the relationship to the flora. The flora changes dramatically to mucin-eating bacteria like Actinomycophilia. I've forgotten the exact bacteria, but you can definitely see changes in the flora, say with the helminth infection. And bacteria then steer metabolism and kind of contribute to these different things. The IgA, and the studies that Barton has done, it's nice to have David Rollet over here, but study that Barton has done, he's back at HHMI now, is important in keeping the commensal flora kind of regulated. And it hasn't really been done carefully enough in the dynamic setting in which we really evolved. So I wouldn't be surprised if there's a relationship between the IgA and the B cells that are IL-5 receptor positive, and the B1 cells that are in serosal peritoneal cavities. And IgA production, that's important, commensal oriented, because many of these recognize these germline sequences that see bacterial conserved sequences in gram-positive and gram-negative. So it's just datapenia. Holly would call it mural dyslexia, right? The handwriting's on the wall, but unable to see it. Dave, have a guitar player.
Was there another question?
Thanks.